right, this is a re-recording of the lecture from November 1st. The original lecture had some audio difficulties, so this is a re-recording. So at the beginning of this lecture, we reviewed what we had gone over so far in mitosis. And so in mitosis so far, we had talked about prophase, right? The first stage here after interphase. The chromosomes are condensed, the nuclear envelope starts to disappear, and those mitotic spindles start to form. That next phase is pro-metaphase. Right? By this phase, the nuclear envelope is completely gone. Right? The mitotic spindle is completely formed and is connected to the chromosomes via the kinetochore. And those centrosomes, where the mitotic spindles are coming out from, are moving to the poles, or moving to the opposite sides of the cell. And then lastly, we talked about metaphase. Right? In metaphase, the chromosomes are lined up on the metaphase plates. Essentially, those mitotic spindles connected to those chromosomes are playing a game of tug-of-war, and they will line up those chromosomes directly in the middle of the cell. All right, so we only got through 3 phases of mitosis, but there are still some more phases to talk about, so we're going to jump right in. All right, the next phase after metaphase is anaphase. All right, in anaphase, we are splitting apart our chromosomes, or we are breaking the connection between our sister chromatids. All right, so either side of the chromosome was linked to a mitotic spindle to an opposite pole. The connection between our sister chromatids have been broken, and either sister chromatid from an original chromosome is broken apart and is going to start to migrate towards the opposite pole. Right? So this is going to happen as these mitotic spindles start to shorten and bring these sister chromatids to either pole. Our next phase is telophase. Right? At this point, the chromosomes, they have reached their respective poles, right? They have reached opposite sides of the cell, and they're going to start decondensing, right? So they're going to go back into that decondensed form, much like we saw during interphase. The nuclear envelope is now going to reform, and what we have is one cell that has 2 nuclei, right? Well, we got to take this one cell and make it into 2. So this last step, right, it's called cytokinesis, and that's where we are breaking this one cell with 2 nuclei into 2 cells, each with their own nuclei. All right, so cytokinesis is the splitting of the cytoplasm, right? And in this point, we are separating our 2 nuclei into different cells. And now, depending on what organism we are talking about, right, different organisms go through cytokinesis differently. So in animals, we get something called a cleavage furrow, right? It's essentially like a drawstring that you might, say if you had a balloon and you put a string around it and you tied it and you kept pulling that string tighter and tighter and tighter, right? That's essentially what is happening in cytokinesis in animal cells. Right? We have this cleavage furrow that is formed. It's being constricted until it finally separates into 2 separate cells. Now in plants, they do things a little bit differently. Right? If you remember, the plants, they have a cell wall that keeps the outside of our cell nice and rigid. Right? And so in plants, they're actually going to form this what's called a cell plate. Right? And it's basically a cell wall that is forming in the middle of this one cell with 2 nuclei, and eventually will divide this larger cell into 2 daughter cells. All right, so in class we went through this group review here. I would encourage you to pause the video and see if you can't go through this again together. All right, I would encourage you to pause it now and then come back and we will go through the answers. All right, so let's go through these answers. Number 1, right? This cell that's circled as number 1 here, right? I would label this one, excuse me, I would label this one as prophase, right? We can see the individual chromosomes, right? They have been condensed. versus the cell over to the side here, I can't see the individual strands of my chromosomes, right? So they have been condensed, but they have not yet been organized, right? So I would say this is prophase. Number 2 here, right, is likely telophase, right? We can see that there are 2 nuclei in one cell. These nuclei have chromosomes that are not condensed, right? We cannot see the individual strands. Now, just the way that this image is taken, right, it looks like these nuclei are overlapping, but remember this is a 3-dimensional cell, so it's just the angle of the image that makes it look like it's overlapping. They are not overlapping in real life. Number 3 here would be best represented by metaphase. We see our chromosomes are lined up on this metaphase plate, right? They're right in the center of this cell. Number 4 would be anaphase. We can see our chromosomes have split apart and are now migrating to the opposite sides of the cell. Number 5, all right, I don't see any condensed chromosomes. Right? This is likely interphase because I only have 1 nuclei and my cells are not condensed. If I look at this image as a whole, I see most of my cells look like this one in number 5 here, right? Because our cells are going to be in interphase most of the time. Number 5 would be interphase. And then number 6, right? It's likely this transition between prometaphase and early metaphase, right? The chromosomes are condensed. They are starting to line up on this metaphase plate, right? But they're not all nice and neat like number 3 was. All right, so it's likely the very end of prometaphase and or early metaphase. Remember, these phases are happening in a continuum. All right, I also asked you to come up with your own mnemonic device, right? And I shared my mnemonic device in class. I like to imagine a puppy that is not yet potty trained and has to use a PP mat. All right, so prophase, prometaphase, metaphase, anaphase, telophase. That's how I remember it. There are a lot of mnemonic devices out there. All right, so we finished up mitosis. Let's move into meiosis, right? And if we're talking about meiosis, we're starting to talk about sexual reproduction. So sexual reproduction is the process that gives rise to offspring that have these unique combinations of genes. All right, so the offspring are not genetically identical to their parents, right? Which is different from asexual reproduction, right? Cells that go through mitosis, some cells might use this in the form of asexual reproduction, and we are giving rise to offspring that are genetically identical to the parent, right? Hydra is a good example of an organism that uses asexual reproduction. We can see it starts to create these individuals kind of budding off of the main body here. It's doing this through the process of mitosis. So these individuals, when they eventually fall off of the main body, right, they are genetically identical to the parent, which is different than a lot of our other organisms, ourselves included, cats, dogs, lots of our pets. They will go through sexual reproduction and we will have offspring with a very unique combination of genes. Now, if we're talking about sexual reproduction, there are some disadvantages and some advantages, right? So the disadvantages is it's complicated, all right? There's more steps to it. There's more going on. It can be costly, right? So you have to invest time and energy to find a mate, and you have to invest into mating strategies. If you've ever seen videos of birds of paradise that are trying to attract a mate, and they might spread their wings and do these funny little dances, right? That's time and energy into a mating strategy to find a mate, right? And there are many examples of this in the animal kingdom. It can be potentially dangerous, right? When organisms are mating, they can be visible to predation or vulnerable to predation. They could also get a disease from their mate. And finally, it's not always successful. We see this a lot of times in news articles about zoos that are trying to breed different organisms that are in captivity, and we might see an article that says, they're trying to breed this tiger and it's just not working. All right, so they might mate, but that mating might not always end up in an offspring. It's not always going to be successful. But there is one very huge advantage here, and that is that we are increasing our genetic variation and diversity between individuals and within populations of individuals. Right? So we're increasing our genetic diversity. Increased genetic diversity means that over time we can evolve and we can adapt to new problems. That means that a disease might not wipe out an entire population with identical genes. All right? We would have some diversity in there, so a disease might affect individuals differently. All right? This advantage is a huge one. It makes all of these disadvantages worth it. All right, so if we're talking about sexually reproducing, we have to talk about life cycles. Now, we're just going to broadly go over life cycles here. If you take Biology 102 in the spring, right, you will go more into depth about these life cycles in different organisms and how they evolved. But we're just going to broadly talk about life cycles, which is a sequence of events that produces another generation of organisms. Right? So for sexually reproducing organisms, this is typically going to involve an alternation between haploid cells and diploid cells. Right? So if you remember, haploid is where you have one copy of your genetic information, as we see in this image here. Diploid is that you have two copies of each type of genetic information. Right? We are a diploid dominant species. Our cells are diploid, right? Unless we are producing gametes, or egg and sperm, and then those cells are haploid. But the majority of our cells will be diploid. All right, so there's three different types: a diploid dominant species, a haploid dominant species, or you could alternate between the two. Right? Most animal species are going to be diploid dominant, like we are. All right? The cells in our organisms are going to be diploid, except for our gametes. Fungal species are a good example of organisms that are haploid dominant, right? So if you see some fungi growing out in the woods, right, a lot of the times what you might be seeing are purely haploid cells because they're mainly made up of haploid cells. Now they do have a diploid stage here, right, but it's very, very small part of their life cycle. And finally, we can alternate between being haploid and diploid. We call this alternation of generations, and many plants will alternate between being haploid and diploid. All right, so if we're talking about sexual reproduction, we have to talk about meiosis. All right, so sexual reproduction requires fertilization. We have to have two haploid gametes unite to create a diploid cell called a zygote, right? So in humans, right, our image up top here, this would be the egg and the sperm. They are both haploid, right? We can see it haploid because it's denoted as N, and it has 23 total chromosomes. So N equals 23, right? Both of these are haploid. Both of these have 23 chromosomes. When fertilization occurs, right, so the egg and the sperm will fuse together to create the zygote, right? When it fuses, we have now created a diploid cell. We have two copies of each type of genetic information. So we could now denote the cell as 2N, right? And now we have 46 chromosomes. So 2N equals 46, right? Now we could also, from this, determine how many homologous pairs there are. We could say there's 46 total chromosomes. Let's divide it by the ploidy level. That means we have 23 homologous pairs. This zygote then would go on to go through stages of mitosis and develop a new individual. All right, so for sexual reproduction to occur, we need the fusion of gametes. Well, how do we get gametes? All right, now that is the process of meiosis. All right, we are going to create our gametes, which are haploid, from an original diploid cell. All right, so broad overview of meiosis here. All right, just like mitosis, meiosis is going to begin after interphase. All right, so after G1, after the S phase where we're duplicating our DNA, after the G2 phase. We can break meiosis into two parts, right? Meiosis 1 and meiosis 2. In meiosis 1, we are going to produce two haploid daughter cells from the original diploid cell. All right, so we're changing our ploidy level in meiosis 1. We're going from diploid to haploid. Right? Any homologous pairs are going to separate, and we're going to exchange some genetic material, and we'll go over this. In meiosis 2, we are taking those resulting two haploid cells from meiosis 1, right? And they're going to go through a series of cell division to create 4 haploid cells, right? And our sister chromatids are going to split apart. All right, so in meiosis, we're going from a haploid cell to a haploid cell. The difference being, instead of one chromosome with 2 sister chromatids, now we just have a singular chromosome in each resulting cell. All right, so let's start by going over meiosis. All right, meiosis 1 tends to have the most differences between meiosis and mitosis. Prophase 1 being a particularly important step where a lot happens. So in prophase 1, our chromosomes are going to condense and our nuclear membrane is going to break down. Right? This is very similar to what happens in prophase of mitosis. However, in meiosis 1, in prophase 1, we get this formation of bivalents, also known as a tetrad. Right? 2 different words describing the same exact feature. A bivalent is when 2 homologous chromosomes come together in a process called synapsis. Right? So synapsis is when these bivalents form, right? And when we have a bivalent, we can get our process of exchanging genetic material through crossing over. Right? We can see these 2 chromosomes here, they have exchanged some genetic material with each other. Segments of these chromosomes are exchanged with each other. So crossing over is super important in increasing our genetic variation, right? Crossing over, it's also called homologous recombination. We are going to exchange genetic information between non-sister chromatids, right? Or exchange information between our homologous pairs. So if we look at our example here, we have a purple chromosome and a green chromosome. They are homologous to each other. They carry the same type of genetic information, right? And our purple one here is going to swap some of that genetic information with our green chromosome, right? So maybe they're swapping the genes for eye color, or they're swapping the genes for height, right? The result will be a little bit of genetic variation because we have added a little bit of our green chromosome to our purple one, and vice versa. Right? So we would call these chromosomes, after crossing over occurs, recombinant chromosomes. All right? So increasing genetic variation. Right? So it's creating new combinations of our parents' genes within our gametes. Right? What it does not do is increase or reduce the likelihood of a mutation. Right? It can also not create any new genes, right? So if these chromosomes did not previously carry the gene for eye color, right, they are not going to spontaneously gain that gene, right? They're not creating any new genes. They're just swapping between the genes that they already have. So let's take another look at this. This time we're going to look at crossing over, and we're going to introduce the concept of alleles. All right, so an allele is one of 2 or more versions of DNA sequence at a given loci, right? Loci is how we say the location on a genome. All right, so basically an allele is a different version of a gene, right? So this would be blue eyes versus green eyes, blonde hair versus brown hair. All right, it's still coding for hair color, still coding for eye color, right? But it's a different variation. And so let's walk through this with a homologous pair here, right? So we have a bivalent or a tetrad that has formed between these two homologous pairs. The red pair, or the red chromosome here, has the allele capital C and the allele capital E, right? The blue allele here has the alleles little c and little e, right? So the C's here would represent a gene, and maybe this gene is eye color, whereas the big C might be brown eyes and the little c might be green eyes. The E's also represent a gene, a different gene, but two variations of a gene. So maybe this gene is hair color, right? Brown hair versus blonde hair, right? When a tetrad comes together and goes through crossing over, they essentially break off segments of their chromosome, right? They essentially pinch together at what's called this chiasma, right? They pinch together, they twist around, and then they give away their information to the opposite pair, right? So now we see instead of an all red chromosome here, we have one side that's red, one side that's red with a little bit of blue, right? Same with this blue chromosome here, right? We have changed the allele that this side of our red chromosome is carrying. Now if we were to take these resultant homologous pairs, these recombinant pairs here, right, and separate them out into gametes, and so each side of each chromosome would end up in a singular gamete, we can see we have variation, right? If crossing over never occurred, we would have two red and two blue, right? But because crossing over occurred, we've increased our variation. We have a red, we have a blue, but we also have two that are red and blue. All right, so that was just the first phase of meiosis here. Prophase 1 has a lot going on, like I said. In prometaphase 1, the nuclear envelope is gone, right? Completely gone, just like we see in mitosis. The spindle apparatus is complete and attached to our chromosomes, but how they are attached is slightly different, right? So they are going to be attached to either side of the bivalent, right? So one mitotic spindle is going to attach to one side of our bivalent. The other mitotic spindle is going to attach to the other side of our bivalent, right? That's different than mitosis. Mitosis, we're attaching a mitotic spindle to either side of a singular chromosome. Or we could also say we're attaching it to one sister chromatid and the opposite sister chromatid. Right? So this is where we vary here. Mitotic spindle is attaching to either side of the bivalent. Mitosis is attaching to sister chromatids. All right, so then we're going to move on to metaphase 2. Excuse me, I keep saying 2. Metaphase 1. All right, metaphase 1, we are going to organize our chromosomes along the metaphase plate. But again, unlike mitosis, we're going to organize our bivalents along the metaphase plate instead of our singular chromosomes. And so our bivalents, when they're organized in the metaphase plate, they almost look like it's a double row, right? We have a tetrad here, we have a tetrad here. Now when they line up on the metaphase plate, this is completely random, right? The way that they choose to arrange themselves, random, completely random. So when we have two tetrads, we have two possibilities of how they might arrange. They might arrange like this possibility 1 here, where both of the blue chromosomes are on either side and both of the red are on either side. Or they could arrange where we have a blue and a red on either side. Completely random, right? This random arrangement is called independent assortment, right? An independent assortment is, again, another way that we can increase our genetic diversity. So if we brought this cell through the end of meiosis 1, right, we see that if they are arranged in possibility 1, right, we see these two resulting cells look very different than if they had been arranged in possibility 2, right? And if we bring it through meiosis 2, right, which we'll go through in a second here, we can see that the 4 resulting daughter cells look very different in their combinations compared to the possibility 2 combinations, right? So we're increasing our genetic diversity. Now this example only has 2 tetrads lining up, right? Think about humans though. Humans have 23 homologous pairs, which means we will form 23 tetrads. If we calculate out all the different ways that they can randomly arrange at the metaphase plate, right, there's over 8 million possibilities, right, or 8 million different results from this arrangement, right? So we're increasing our variability, our genetic variability. All right, our last 2 phases here are more or less similar to mitosis, right? In anaphase 1, right, we're splitting apart our homologous pairs. So either side of the tetrad will be split apart and start to be brought to either pole, right? Different than in anaphase of mitosis. Anaphase of mitosis, we're splitting sister chromatids. In anaphase 1, those sister chromatids are staying together and we're splitting homologous pairs. In telophase 1, we're going to reform the nuclear envelope around the chromosomes that have migrated to their respective poles. And then finally, cytokinesis, we will create 2 cells, right, that are now haploid, different from the parent cell, which started off as diploid, right? And these 2 cells will go on to meiosis 2, right? So, again, big picture here, meiosis, we are increasing our genetic variability, and we're doing this in a couple of different ways, right? First of all, we are combining the genetic material from 2 parents, right? We have homologous pairs, one that we got from mom, one that we got from dad. We're combining that genetic material together, right? We are independently assorting those chromosomes during meiosis, right? So how they arrange on the metaphase plate. And finally, we're getting recombination, or crossing over occurring, which also increases our genetic variability. All right, so that was meiosis 1. After meiosis 1, we get those 2 daughter cells that are haploid, right? We are going to immediately take those 2 daughter cells and move it through mitosis or meiosis 2, right? There's going to be no interphase in between meiosis 1 and meiosis 2. All right. Now the events in meiosis 2 are so similar to mitosis that I'm not going to go through each slide of a phase, right? But rather we're going to talk about it all on this slide here, right? Because they're so similar to mitosis. So we'll start with prophase 2, right? We have a haploid cell. We see that there are no homologous pairs. A good indication of this is that I have a chromosome that's large and a chromosome that is small, right? They're not homologous to each other. They are not carrying the same type of genetic information, which is why this cell is now haploid with 2 chromosomes. All right, prophase 2, these chromosomes, they're still condensed, right? Our nuclear envelope is going to start to go away, and our mitotic spindle is going to start to form. By the time we get to prometaphase 2, which is not shown in this image here, right, the nuclear envelope will be completely gone. The mitotic spindles will be completely attached to our chromosomes. And just like in mitosis, that mitotic spindle is attaching to either sister chromatid. All right? So, because it's attached to either sister chromatid here, in the metaphase plate, we have just singular chromosomes lining up, instead of 2 double rows, right? We just have a singular row. Our individual chromosomes are lining up. Anaphase 2, we are splitting apart our sister chromatids, and they are going to either side of the cell. In telophase 2, we are reforming that nuclear envelope. All right, so we have a cell with 2 nuclei. Remember, this is happening twice, once for each cell that was originally created in meiosis 1. And then finally, we have cytokinesis, right? And we are forming our gametes at this point, right? They are haploid. They only have 1 chromosome, right? And they are ready to go on to be used in fertilization. Now, a point I want to make here is that in meiosis 2, we're starting with a cell that is n equals 2, or it's haploid with 2 chromosomes, and we're ending with a cell that is n equals 2, or haploid with 2 chromosomes, even though they look differently, right? So remember, when they're in this X shape, that just means that either side has gone through DNA duplication, right? We see a little bit of color changes here because of recombination that had already occurred, right? But it's the same genes on either side. Might be different variants of the genes, but the same genes, right? When we get down to the end of meiosis 2, right, it's just an individual chromosome that does not have a duplicated portion to it, right, because we no longer need it. But they are each still a chromosome in both of those 2 cells. All right, I like this image. It shows you a great comparison between mitosis and meiosis. Again, mitosis, we are producing 2 diploid daughter cells. So we're going from a diploid cell to a diploid cell. And those daughter cells are genetically identical. There is no crossing over occurring. In meiosis, right, we are going from a diploid cell to 4 haploid daughter cells, right, that have some genetic variability to them. All right, we talked about these questions in class, and we will start our next lecture with these questions as well. So I would encourage you to work through these questions, and we'll review them when we come back together.